Hi everyone, this is Jonathan Cedillo, student at Grand Canyon University, and today I'm presenting to you corticosterone, vasopressin, and oxytocin influence on anxiety. Presentation for the class Biology 515, Concepts of Human Physiology 1, Professor Sandra Barrera. Background. In the following paper, social isolation rearing induced anxiety in response to agomelatine in male and female rats, role of corticosterone, oxytocin, and vasopressin. The relationship between the suppressing action in anxiety of agomelatine and cortisol was studied, together with the influence that vasopressin and oxytocin have on this activity, and if sex dependent effects are seen. Agomelatine is an antidepressant agent that can cause phase adjustment of the body clock. In this study, scientists have demonstrated signs of anxiety in adult rats and reduction in corticosterone concentration of rats that have been isolated. Agomelatine drug was used to treat anxiety, which is known to be an anxiolytic. But in this study, it did not increase levels of lost cortisol, meaning that rats acquired hypocortisolemia. In addition, this study explains that oxytocin is an anxiolytic agent and vasopressin is an anxiogenic agent, which are related to circadian disorders. And they also showed evidence of being sex dependent differences in response to particular circumstances. In this study, scientists describe agomelatine as an antidepressant, which is dependent over the activity of melatonin MT1, MT2 receptors and acts as an antagonist of serotonin 5-HT receptors, which are mostly present in the suprachiasmatic nucleus of the hypothalamus. This antidepressant is also said to be an agent that corrects circadian rhythms. Authors of the paper state that an adversity in life is anxiety that might be caused by the isolation of individuals as presented in the experiment with rats. Authors also reveal the effects of the medication over this neurodevelopmental model of anxiety. The experiments show that late life stages of the rats were presenting symptoms of anxiety of both male and female rodents, while also showing reduction of corticosterone levels in males and females. Studies confirm that low levels of this hormone is related to anxiety development, which is said to be related to show suppression of the hypothalamic pituitary adrenal axis. Interestingly, authors of the paper reveal that even with agomelatine treatment in rat models, the corticosterone low levels did not increase. This is only suggesting that corticosterone is not an agent that causes anxiety and that agomelatine anxiolytic activity affects other pathways. Oxytocin and vasopressin. Continuing, the article explains that vasopressin and oxytocin are related substances in anxiety, stress coping, and social grouping. The paper also explains that central oxytocin acts as an anxiolytic and antidepressant. On the contrary, vasopressin acts as an anxiogenic and depressive agent. These neurotransmitters disorders in circadian rhythms can present anxiety events, especially in the case of vasopressin. In addition, the neurotransmitters have different effects depending on gender. However, there is insufficient data in female models, which represents a problem taking into consideration that stress-induced mental disorders, including PTSD, are more common in female than in males. During anxiety, oxytocin produces sex-dependent effects over the activity of the amygdala, while for vasopressin, prenatal stress-induced social memory deficits are more prone in female rat models. While vasopressin 1A receptor in mRNA synthesis was not affected in males, in the contrary, it lowered its expression in females. Additionally, an induced depression model was used to show the transcriptional modifications in the hypothalamus influenced by the stress, by the sex-specific and ovarian hormone dependency. Authors of the paper state that agomelatine's activity in restating the circadian rhythms also have a positive effect in circadian hormones, such as cortisol and melatonin, and that it provides anxiolytic effects. With this, scientists ask themselves if vasopressin and oxytocin might underlie the earlier observed anxiolytic-like actions of agomelatine in the rat models, perhaps more so than corticosterone, and whether, if any, sex-specific effects are evident. Materials and methods. For this experiment, the article explains that 72 rats were used. 36 males and 36 females. Then the rats were isolated in either one model per cage or three models per cage. The completely isolated rats and groups of three rats were treated with the drug agomelatine, which was administered intraperitoneally. Scientists explained that injection administration of agomelatine was conducted during afternoon hours due to its effects are best approached during that time of day in rodents. After treatment, rats were sacrificed and blood samples were collected to be analyzed, targeting the substances of interest, which are corticosterone, vasopressin, and oxytocin. Next, the article is presenting and explaining the results about the effects that the neurotransmitters of interest have when the animal models were isolated. The isolation demonstrated an anxiogenic effect on both male and female models, revealed as decreased percentage TOA or TOA and hypercortisolemia. The drug treatment inhibited anxiety symptoms in both rat sex, but there was no effect in the acquired hypocortisolemia. Scientists analyzed the percentage TOA, corticosterone, vasopressin, and oxytocin data. Here, Table 1 shows a correlation between oxytocin, vasopressin, and corticosterone, and percentage time in open arms, or percentage TOA. In all treated individuals and groups, the table presents a two-way ANOVA analysis, which shows not compelling gender treatment reaction on corticosterone, but results also explain that there was an effect in corticosterone levels which depend in the sex of the rats, together with a compelling treatment effect. Additionally, the two-way ANOVA presented no compelling gender treatment reaction on oxytocin and vasopressin. In contrast to cortisone, the rat sex had no compelling activity on oxytocin or vasopressin. Interestingly, the drug treatment had a significant main effect on oxytocin and vasopressin. Here, in Figure 1, the paper is explaining the levels of the neurotransmitters and the hormone in the rat models. Corticosterone in graph A, oxytocin in graph B, and vasopressin in graph C. The groups consisted in socially reared animals receiving a vehicle treatment and social isolation reared, or SIR, animals receiving vehicle and agomelatine treatments. With the graphs presented by the article, we can conclude that the rats in social isolation rearing decreased corticosterone in both sexes dramatically. And we can also observe that agomelatine did not reverse hypocortisolemia, as shown in Figure 1a. In addition, social isolation rearing drastically downregulated oxytocin in both sexes, as presented in Figure 1b. But it did upregulate vasopressin in both sex models, presented in Figure 1c. The drug treatment did downregulate vasopressin concentration in both sex models, shown in Figure 1c. Interestingly, agomelatine treatment also upregulated oxytocin concentration in female only and not in males, as shown in Figure 1b. Analysis to compare the agomelatine-treated female and male rats data was put together. However, no significant data was evident from the groups. Further, only noteworthy negative interrelationships were identified between vasopressin and oxytocin, and between vasopressin and corticosterone. But noteworthy positive interrelationships were identified between oxytocin and corticosterone. Taking into consideration the neurotransmitter-anxiety relations, vasopressin concentration and percentage TOA demonstrated a compelling negative correlation, which is shown in Figure 2. Also, in the same figure, oxytocin and corticosterone did not show a compelling relation to percentage TOA. Discussion. Life stress situations can bring anxiety issues to individuals. 5-HT1A and 5-HT2A receptors are synthesized from early stage of life development, but these can be wrongly expressed depending in the situation of individuals. In social isolation, these receptors might be compromised. However, there is treatment to alleviate the function of these receptors, helping to treat anxiety. Additionally, according to this study, circadian rhythms are also compromised during anxiety, but agomelatine drug therapy offers a way to fix disorders related to circadian rhythms. In addition, vasopressin is also related to anxiety problems and social isolation, can upregulate this neurotransmitter and at the same time downregulate oxytocin levels. But in this paper, authors explain that in all treated animals, the oxytocin and corticosterone levels are insignificant to anxiety. Also, it has been already stated that isolation of rats induce anxiety-like behaviors, reducing oxytocin and corticosterone, but elevating vasopressin. Interestingly, agomelatine drug can reduce vasopressin levels in the organism. Further, 5-HT receptors are involved in the delivery of the previously mentioned substances. But again, this receptor is compromised during isolation. Finally, the creators of this literature point out the relation between sex and corticosterone according to the social interactions of the models. However, there was not significant correlation between gender, oxytocin, and vasopressin. This can explain the reason behind why women are more prone to anxiety and depression and men are more prone to being antisocial. In conclusion, the drug agomelatine regulates levels of vasopressin in both sex models, but only regulates homeostatic levels of oxytocin in females and not in males. Oxytocin and vasopressin have different roles in males and females, and their effects may vary depending on the sex. Conclusion: all data presented and explained in this paper is suggesting that social isolation is an anxiogenic causative agent, reducing corticosterone and oxytocin, but increasing vasopressin and altering corticosterone levels depending on gender. Despite these events, both genders showed correlation between anxiety and upregulation of vasopressin. However, agomelatine treatment demonstrated regulation in vasopressin levels in both male and female models. This same event occurred for oxytocin, concentration in a lower proportion and only in females. Corticosterone did not suffer any effect after drug treatment. The regulation of the neurotransmitters, oxytocin and vasopressin, can be accommodated by direct and indirect chronobiotic events. Thank you for watching and see you next time.